Very exciting to be here. It's my first Pi data meeting, so we'll see how it goes. So my name is Julien Cohen-Haddad, and a little bit of background. I'm a PI, principal investigator in a lab, in a neuroimaging lab at Polytechnique Montreal. So our research is focused on MRI technologies, magnetic resonance imaging, for neuro application. So we are based on the University of Montreal campus. And of more interest to you, we are also developing and maintaining open source software for analyzing medical image data, mostly coming from MRI, but also from histology. And lastly, we are also engaged in collaboration with clinicians in order to translate those tools, those open source tools, get feedback and improve our developments. And some example of pathologies include multiple sclerosis and Alzheimer's disease, for example. So I made that slide like 2 minutes ago because I realized that this is a Pi data meeting, and in fact I will mostly talk about data. But the software that I will talk about that we developed are Python based, which is why I was very excited to come here and to meet with the Montreal Python community. So before talking about data, let's talk about and analysis. Let's talk about how those data are acquired at the first place. So how does MRI work? So to get an MRI image, you put someone in the tube, then you slice the person. To be more precise, in fact, you slice that person virtually using MRI gradients and RF pulses. So very briefly, with MRI, the spatial location of spins of each water molecule is encoded in frequency. So the signal you record is actually the 2D Fourier transform of your image. So once you collect all your data, you can apply an inverse Fourier transform and then get the image that looks more familiar to you. So now let's look into what we are really interested in, what our lab is focused on, which is the analyzing, like doing in vivo histology with MRI. So if you look inside the brain at the microscopic scale, you'll see that there's a lot of different structures, and including neurons, which are the structures we are interested in. So this is an example of a histology at about 20 micrometer resolution. However, when we do MRI, the voxel size, so a voxel is a 3D version of a pixel. When you do MRI, the voxel size is on the order of hundreds of micrometers. So you cannot really see neurons or relevant structures in there. So why is MRI getting all the buzz about being able to be quantitative about the microstructure? Without going into too much detail, I can just tell you that MRI is in fact very sensitive and specific to tissue microstructure. There's a lot of research going on that has been going on for the past couple of years. Thanks to advanced technique, we can in fact interrogate things such as the density of axons or neurons in the central nervous system. You might have heard about functional MRI to measure neuronal activity and metabolism that can be quantified using MR spectroscopy. So ultimately, all those techniques are used for improving diagnosis and prognosis in different diseases, and also provide objective biomarkers for pharmaceutical companies and researchers to test new drugs. The problem with this fancy quantitative MRI data is that they require an extremely complex processing pipeline. So that's where the data comes in. And those like pipelines are often executed manually and suffer from poor reproducibility. And examples include, like examples of tasks include feeding, biophysical models, image segmentations, registration, surface reconstruction, and so on. So that contributes to a lack of replicability and transparency that in turn contributes to the neurosceptic trends that we've been seeing for the past couple of years in specialized and popular media. So how do we solve this? Well, deep learning can come to the rescue. It can be leveraged to achieve some key tasks, including segmentation of healthy structures, as well as pathological structures, and categorization tasks for the purpose of diagnosis, for example. And in fact, deep learning is taking on very rapidly in the medical imaging community. As an illustration, the main MRI conference has seen an exponential increase in abstract submissions for the past couple of years. So I'm going to show you some examples of ongoing projects in our lab, and if you're interested, we'd love to receive your contributions, feedback, and so on. So the first example is showing how we can apply deep learning to improve prediction in patients suffering from amyotrophic lateral sclerosis, or ALS. So this disease is characterized by an atrophy of the motor neurons in the brain and the spinal cord. And we can actually delineate the gray matter from MRI scans in order to measure, to quantify atrophy. So in this study, we developed a tool based on deep learning to do this segmentation of this yellow shape, this butterfly shape. I don't know if you see my cursor. No, you don't. And so we showed that this biomarker is more effective to discriminate ALS patients from healthy controls, and then look at their one-year follow-up in order to have a better prediction of those patients. So in the next slide, I'm going to show you the details of the segmentation algorithm. So that has to do with the segmentation task using deep dilated convolutions. So UNet, as you know, is a popular method for segmentation tasks. However, it's sometimes high-level features are noisy, and most of the information is contained in low-level layers. Is something going to explode here? So I think we're fine. So the idea here would be to reach higher levels with less convolutions. So that could be achieved with a true spatial pyramid pooling that add holes in the convolution kernels, and that provides a parameter reduction while increasing the effective receptive field. So we demonstrate the utility of this approach in a public database of spinal cord MRI data. And we also show that the same architecture can scale very well in high resolution images. So here you're seeing an example of MRIs acquired at at hundreds micrometer resolution, so across almost 5,000 slices, and all the slices were segmented automatically with only 10 slices that were required for the ground truth segmentation. So you can find more details in these publications. So now I'm going to show you a follow-up project on deep supervised, semi-supervised segmentation with weight average consistency targets. The main problem in medical imaging is that we have, sometimes we have tons of data, but we have very few annotations, because those are usually done by radiologists. They are busy, and it's very difficult to get a reasonable amount of data for training models. So the main idea here was to extend the semi-supervised learning with mean teacher method to a segmentation task. So let's see how it works. In the mean teacher method, two models are trained in parallel. Let's see, I can bring my cursor here. The student model, which is here, learns in a traditional way with inputs and associated labels. And the teacher model, which is here, receives unlabeled data. So we don't need to provide labeled data for this part of the model. So along with the segmentation loss, which is the dice here, we update the weights of the student model. And for the teacher model, the weights of the model are obtained by averaging the weights of the student model across previous training steps. So over time, the prediction of the teacher model will be more accurate than the teacher model, than the student model itself. So the main trick of that paper was, compared to a classification task, was to make sure that for the unlabeled data, we have the same transformation for data augmentation that is applied on the image before entering the student model. So for the network architecture, we used a UNet with 15 layers. And so I'm not going into details for the training parameters. You can find more information on those on the paper here. But results overall show a better performance of the semi-supervised approach when compared to an architecture with less data, where less data are seen by the model, even if the ground truth is missing. Another follow-up that we did from this study, which was a natural follow-up, is we wanted to address the issue caused by having a mix of data from different centers. So usually data come not only from one clinic, but from different clinics, and the image looks slightly different. So the idea here was to combine the mean teacher method that we saw previously with an unsupervised domain adaptation approach proposed by French et al. So briefly, the domain we want to adapt, our model 2, has unlabeled data, and they are introduced in the same way as for the mean teacher model with a single domain. So the unlabeled data from the other domain is augmented and then fed to the student model in the same way as we saw in the previous slide. But then the weights of the student model are updated, and the teacher model, the weight of the teacher models are updated in order to perform in a more in a better way than the student model. So for the results, we had 4 different centers. We trained the model on center 1 and 2, evaluated on center 3, and tested on center 4. And results overall show an improvement that are specific to the domain adaptation. We made sure that the improvements do not come from unlabeled data by doing some ablation experiments. Again, you can find some more details on the publications. So let's move now to another application, which is very hot at the moment, and it's basically consisting of mapping multiple sclerosis lesions in the central nervous system. There's already a couple of algorithms that exist for doing automatic detection and segmentation of those lesions in the brain, but so far none have looked at the spinal cord. So that's what we wanted to, that's one problem we wanted to tackle. So we teamed up with 12 of the biggest MS clinics in the world that shared data from 600 MS patients. Also 8 experienced neuroradiologists who provided the ground truth segmentations. Image processing was performed using Spinal Toolbox, which is a Python package developed in the lab. And one of the big challenge of this is that there is a huge class imbalance. Typically, an MRI scan has 128 by 128 by 60 voxels. And an MS lesions represents only 10 to 50 voxels. So there is a very large class imbalance that is challenging for the model to learn. So in this method, we proposed a cascade of convolutional neural networks. The first CNN detects the spinal cord, so we can crop around it. And then there's a second CNN that takes care of the MS lesion segmentation in the vicinity of the spinal cord. So this algorithm has been implemented in Python, and results show robust results depending on the image contrast, orientation, resolution, and orientation, and so on. Fairly good sensitivity, but we have quite a large number of false positive rates. So basically, lesions, like voxels are labeled as lesions, but in fact it's artifacts or not lesions. So there's still room for improvement, but so far we've implemented that model and that framework into SCT, which is our package for analyzing spinal cord MRI data. And this has notably been featured in the NVIDIA newsletter 2 years ago. So we're quite encouraged by this. But to sum up on what we've seen so far, and to maybe discuss about the outstanding issues. As I mentioned, there's a low amount of data, and especially manual labels in medical image. And that is partly related to issues with ethics and data privacy, especially here in Quebec. There's also a variety of inputs, image resolutions, image contrasts. So it's very difficult to set proper batch size when we train models. Because ideally we want the model to work for all the existing MRIs on the planet. When we train with multiple contrasts, sometimes data set might be missing, so it's a bit tricky. Published models validated in well-curated single-center data usually fail when they are applied to other centers. So you can see when you read the literature, you see many publications that claim that they have like a dice of 95%. It's amazing, and then when you try the model on your data, it doesn't work because the data come from another domain and it basically does not generalize well. And apart from that, it's not related to deep learning specifically, but the code is rarely publicly available. And when it is, the maintenance, commenting is often of poor quality. So it's difficult to basically rework an existing code or use it. So when we look at the problem we are facing, in fact, people are different, but not that much. So when we look at MRIs of different people, they look pretty much the same. But the real problem is that if you take the same person that you image with different MRI parameters, different field of views, and so on, this is where the biggest data variability comes into play. So what are the possible avenues? You might have heard about generative adversarial networks, or GANs. They can be used to generate synthetic domain, and each new domain would then be mapped into a synthetic domain to perform any tasks, such as segmentation, categorization, or so on. There are several problems with GANs. First is that image features not only varies between centers, but also across parameters. So it would be difficult to create a synthetic, to basically find a transformation that would fit all the existing differences across the MRI parameters. Another problem is that domain adaptation can introduce some bias, as Joseph Paul Cohen showed in a recent paper. Another aspect that we absolutely need to consider is that when we do MRI, we usually have several contrasts, what we call T1 weighted or T2 weighted. So the image looks different depending on the parameters. And we should ideally use all the available contrasts to improve the robustness of the deep learning task. And in fact, this is what a radiologist is doing. When a radiologist looks at images, he's not only looking at one contrast, but several contrasts on different screens, and then perform the diagnosis. Lastly, another cool approach would be to add priors from MR physics in order to generalize models better. So we know that some parameters, such as repetition time, flip angle, and so on, do affect the image contrast. So why not introducing them in the model training so the model would ultimately learn how these parameters affect the image contrast. So this is something we're currently working on, and I'll show you some preliminary results. So the research program that I'm going to talk about is funded by IVADO, which is a program put together by the University of Montreal and done in collaboration with Joseph Paul Cohen at the MILA. So first, we looked at the film architecture. So film stands for visual reasoning with general conditioning layer. So in other words, it learns to adaptively influence the output of a neural network by applying an affine transformation to the network's intermediate features based on some input. So in the original paper, film was demonstrated for visual and language tasks, And in that case, we'll see how we can adapt it for image segmentation tasks. So we have an input image, okay, a standard convolutional neural network, and prediction. In that case, would be a segmentation. So let's see how this architecture would be modified with film. Here, some of the outputs of some layers would be modified by an affine transformation. And this affine transformation is defined by parameters such as beta and gamma that are generated by a film generator, which is a multi-layer perceptron that takes as input MRI parameters or contrast, and then optimize those gamma and beta. So during training, film learns to adapt its affine parameters based on the image and labels that it sees. So our hope here is that film would learn how the image contrast is generated by the various MRI parameters that exist. So this would ultimately enable us to create models that can generalize to all the different MR contrasts in the world. It's a pretty ambitious goal, but yeah, let's see where we got so far. So obviously we need data, and we need data from various centers. So we led an initiative with 40 different sites around the world that basically shared data, and each of these sites have a different MRI like model and vendor. Each of these sites acquired data in 6 subjects, so 3 males and 3 females, 7 contrasts, 15 slices. So we ended up with about 25,000 2D images with labels. And the good news is that this data is open access. So you can click on this link, and then you get 25K of MRI data with the labels. So let's see how film would then work with that. So for the training, we used 144 subjects scanned at 24 sites, and we trained it on 5 different contrasts. Okay, so you can see that the contrast look different, whereas even though we are looking at the exact same structure. Okay, it's basically an axial view of a cervical spinal cord. For the testing, we used data from 24 subjects scanned in 4 unseen sites with 1 unseen contrast. And results when comparing units with the film layer show a slight improvement for the film approach. Although we have to be fair, we are quite hitting the limit in terms of inter-rater variability at such high dice regime. So we are not convinced that we see a specific improvement of the film. So there is still some potential that we are currently working on. And one of the approach we are currently looking at is taking into account the MR physics, because the problem is that film, when you specify MR contrast, it cannot necessarily generate across the variety of MR contrast that can exist in the world. Ideally, we would like to generalize across the acquisition parameter space. So we know from the MR physics theory how the signal is related to the acquisition parameters via the Bloch equation. So that's an equation. That's an example of an equation where the signal S can be explained by a good portion of variance by the MR parameters. So in green, there's the parameter that we know. These are the acquisition parameters. In red, it's something that we don't know, but we could potentially constrain, or the model might be able to learn. And these are dependent on the tissue. So it's kind of a chicken and egg problem. We are trying to infer things on the tissue that we actually don't know yet. But our hypothesis is that by introducing the MRI equation, we might be able to regularize the signal and the interplay between all the input parameters, possibly generalize better. One of the problems we are currently having, and maybe some of you have brilliant ideas that can help us, is how to introduce those kind of equation in the architecture while training the model. So that was it for the MRI part. Now let's come back one step back to our big brain here. This is our big voxels that we acquired with our fancy MRI techniques. And one question that many researchers have is, how accurately does MRI measure represent the actual reality of my tissue? So one way to answer that question is to basically reverse engineer that by using histology. So we would basically take the same tissue and do histology on that with high resolution techniques. So how does that work practically? So I'm going to show you an example of workflow that we implemented in the lab. So first you take some tissue, like a brain, for example, an ex vivo brain. You put it into the MRI. You acquire your fancy MRI techniques. Then you take your tissue, same tissue, important, and then you prepare it for histology. So staining with osmium, for example, embed that into epoxy, polishing. Then you use your fancy electron microscopy hardware, and then you basically acquire like small patches of your tissue at very high resolution. We're talking nanometer resolution. You patch, you merge, you stitch all your images together, you segment your image, you resample at the MRI resolution, you do some nonlinear registration, and then you can finally do your pixel-wise correlation in order to compare your MRI values in ordinates versus your histology values in the abscissa. So that's in like 1 minute how the workflow works. But wait a minute, I went a bit fast on one step, which is a critical step, which is to segment the tissue. So let's spend a few minutes on that step. If you take a full-scale image of, let's say, a rat spinal cord tissue, that's how it looks like. When you start zooming in, you have the scale at the bottom left of each panel. You start seeing the microstructure that we are interested in. So what we want to do, our goal here is to segment individual neuronal cell called axons in order to then compute the metrics that we are interested in for validating our MRI data. So let's put that into numbers to see how much work that represents. So let's say a density in the central nervous system, density of neuronal cell is about 100,000 axons per millimeter square. The white matter cross-sectional area is about 10 millimeter square. The time to segment one axon plus myelin for a well-trained grad student is about 1 minute. So the time it would take to segment that particular slice here would be about 2 years without eating, sleeping, and so on. So that's a fantastic application for deep learning, of course. So we've developed another package called AxonDeepSeg, which essentially includes different UNet models trained on scanning and transmit electron microscopy to do this kind of segmentation work. So the details of the implementation and results could be seen on this on Aldo Zaimi's paper. And here I'm just showing you some examples of ground truth in grayscale and the prediction, the inference that this AxonDeepSeg gives you. So in blue you have the axon, and in red it's the myelin surrounding the axon. So it kind of works for mouse transmit EM, human scanning EM. And when we apply that to large-scale image, so the code can handle multi-patch data, overlapped windows, and so on. And then it can basically save a lot of time to the poor grad students who previously had to manually label all those axons and myelin. And then from that kind of output, we can really quantify useful values to compare it with MRI. But we face, again, the same problem as with MRI, is that when we launched AxonDeepSeg, we had maybe 1 or 2 users with similar acquisition parameters that what we used to train the models. But then for the past 2 years, we've had more and more users from all around the world, and they were trying fancy, weird acquisition parameters, and suddenly when they are trying AxonDeepSeg with a trained model, it would miserably fail. And that's because of the heterogeneity across the acquisition parameters. So users have very specific needs, in fact. When they set up a series of experiments with the same tissue type and a fixed set of acquisition parameters, usually the images look very similar. However, when they start changing the acquisition parameters, look at different tissues, brain, brainstem, the heart, then suddenly the models perform very, very poorly. So our strategy here is less ambitious than the strategy we had for MRI. We are not trying to generalize and find a model that fits all needs. We are basically trying instead to provide a framework so that users can easily train a new model for their application. And of course, the main question then is how to minimize the user's efforts in creating those labels, those ground truth. So one way to do that is to do active learning, which is a subfield of deep learning, where the idea here is instead of randomly selecting patches to annotate the training for basically the ground truth, here we would select patches that are most uncertain at prediction time, and we would then fix them, retrain the model with these added patches. So the workflow looks like this. We have a training set with an existing model. We train the model, and then we select the most uncertain inference results. And this uncertainty can be defined through different ways with Bayesian framework, but here we use Monte Carlo sample of models with dropouts at test time. So we pick this bad prediction, and then we manually fix it, which usually takes less than 1 minute. And then we add this fixed prediction as a new ground truth for retraining the model. So here are the results. In Ordinate, you have the dice on the testing set, and on the abscissa, you have the number of iterations. And at each new iteration, there is a new ground truth patch that has been added to the training data set. So it's at each new iteration, basically the model improves. But what's interesting is that in blue, there are the results when patches are randomly selected. Whereas in green, this is the result of when patches are selected using the uncertainty information. So it shows that after only 2 to 3 iterations, the performance of the model almost plateau. So overall, it's encouraging. There's obviously room for improvements. We are looking to combine that with the mean-teacher approach, for example. But overall, that's quite an exciting avenue. All right, so in conclusion, large databases are exciting, but small data sets with few labels is the reality for most medical applications. So we need to find concrete solutions for that. It's really like a scaled problem compared to the ImageNet, or we're not dealing with millions of images and labels. Here we are really dealing with hundreds or thousands of data. There's obviously this quest for generalizability, and depending on the context, we should ask the question whether it is better to provide several application-specific models or one model that fits all applications. So in my presentation, I showed you 2 different strategies. The ultimate perfect model for MRI that we are still trying to get, and the more personalized models for histology. So I think in the field of medical imaging, real impact can be made if good solutions are provided for such scarce data, and code is shared, well documented, and maintained. And lastly, how do you get brilliant ideas in this very like competitive fields? Innovative solutions are usually found by bringing communities together. So that's partly why I'm here. I'm trained as an MR physicist with a bit of a medical background, but I dived into software engineering because it's key to be able to bring together the field of physics, computer vision, software engineering, deep learning. And what's exciting in Montreal is that all those expertise are already present and vibrating. So I'd like to thank everyone in the lab. We have open positions. If you're interested to master's, PhD, or even work as a software engineer for some of the software we develop, shoot me an email. We thank our collaborators who shared data and also collaborate on some of the projects that I showed you, as well as funding sources. And thank you for your attention.